So I don't remember exactly what I've been saying, what the last thing I said here was, but the whole point of this slide is the labeled line code. And that is the notion that taste buds taste all of the different sensory modalities, and there are individual sensory neurons in each taste bud that are attuned to each of the different taste modalities, being sweet, umami, salty, sour, etc. And then the brain interprets the signal coming from that neuron because the line that it's coming in from, the brain interprets any signal coming along that line as being salty or sweet, etc. And it converges on the gustatory cortex. Now, I will point out something here. That little area, I could probably have pointed to any area in the brain. It wouldn't have made much of a difference to any of you. I'm not expecting you to be familiar with neuroanatomy, but I will say that the podcasts that we listened to last week talked about how the different sensory modalities interact with taste, and in particular, they focused on hearing sounds, how sounds affect it. And I'll point out to you that this gustatory cortex right here, this little lobule, it's called a gyrus, this gyrus of the brain right there, immediately below it. So if your brain looks sort of like my hand here, my fist, this thumb part of my fist is called the temporal lobe. It's sticking right below that gustatory cortex there. The temporal lobe is your auditory cortex. So your gustatory cortex and the auditory cortex are direct next-door neighbors. Those two parts of the brain are directly adjacent, and so projections from one part of that brain to the other are relatively easy to establish, and it's not surprising that there is a connection between those two sensory modalities. I'm just giving you a little bit of the neuroanatomical underpinning of the connection that they were talking about experimentally in those podcasts. All right. Yeah, I should probably highlight that in that slide. Okay, so I guess I didn't make a big deal of it on that last slide, but that rainbow line that was heading up towards the brain, there were actually two specific nerves that were represented by that. There was the glossopharyngeal and the facial nerve. And the point is that there are two totally separate taste and salivation circuits in the body. You have two sort of different tongues and salivatory gland pathways in your body. So first, let's define a couple words. Efferent. Let's start with afferent. A on the right hand side by the tongue. Afferent is anything that is sensory. So like affect something is something that has come into you. I have been affected by this. It is a sweeping change. The election affected me, or whatever. It was something that came into you. And then to effect something is outward directed. You effect a change. So that's outward. That means, in this context, afferent means signals that are going up into the brain, sensory signals going up into the brain. And then efferent signals are signals that the brain has processed that sensory information, and then it's going to send signals out, motor signals out to the brain. Motor not just meaning muscle, but it can also mean any kind of effector. And in this instant, your salivary glands, the glands that are going to make the saliva that will have the enzymes that will help digest the food. So you put some food on the tongue, you get these taste sensations, and those sensations ascend via the afferent nerves to the gustatory cortex, and then the brain sends these efferent signals downward to the different salivary glands? Well, the tongue is divided in half, front to back, and along this thing called the sulcus. You can, I guess I probably should have had you guys next year when I rethink the sequence of all this material and what stays and goes. When I had you counting each other's taste buds, I probably should have had you look way deep in there at each other's tongues, because you can see the sulcus. It's this sort of groove. It's a bit of a valley it's kind of far back towards the tongue. On the tongue, you really have to stick your tongue out. It helps with the tongue depressor to see that sulcus. But the taste buds on the anterior or the front side of that are served by what's called the facial nerve. It's a cranial nerve. There's two ways that nerves get in and out of the brain. They either get in and out via the spinal cord and spinal nerves, or they get out via the cranial nerves. Cranial nerves don't come off the spinal cord; they come directly off the brain. And the facial nerve is one of the 12 cranial nerves. It's cranial nerve 7, in fact. So cranial nerve is bringing sensory information, taste, from the front of the tongue up to the brain, and then the facial nerve carries efferents stimulation or signals down to the salivary glands on the anterior portion of the mouth. And these are the sublingual and the submandibular glands. So sublingual, lingua means your tongue, and mandibular, mandible, is your jawbone. So the sublingual glands are right underneath the angle of your chin in the soft part under there, and the submandibulars are just a little bit further back underneath the corner of your jaw and the soft part of your throat. So that's the first circuit of afferent-efferent gustatory circuit, all right? And then a totally separate pathway. Did it evolve? What are the evolutionary origins? I don't know. This is a question that I have raised myself. I would like to know what the evolutionary origins of these two separate taste paths are. How far back does it go down the phylogenetic tree? Why are there two separate pathways? I can't answer those questions for you. They're intriguing to me. And I'm not even sure if there is a good answer, but there may be, phylogenetics being what they are now. So the posterior part of the tongue is served by something called the glossopharyngeal nerve. Glossus is another a term referring to oral anatomy, and pharynx is the posterior part of the throat. So it's really the back part of the throat. The glossopharyngeal nerve carries sensory information from the back part of the tongue. If you look at it, it's much more stippled. It has much larger taste buds than the anterior portion. And then it goes down to the parotid gland. And the parotid gland is a salivary gland that's sitting on your face, really, by your ear, just below your cheekbone and back by your ear. It's in this soft part right below your cheekbone. It's the part of, I don't know if it happens to everybody, but when I suck on a lemon or something like that, my parotid gland starts shivering, right? Kind of does that. It's the parotid gland back there. And it transfers saliva to the back of the mouth and the throat area via the parotid duct. Now, why do we have so many different salivary glands? Turns out each of them makes a slightly different type of saliva. All right, there are two categories of saliva. There's serous saliva and there's mucus saliva. The sublingual, the one right in the front, is more mucosal. The ducts, if you ever lift your tongue, look underneath your tongue, you'll see two little dots along the lingual frenulum, which is the tag that holds your tongue down. There's little dots on either side. That's where the saliva comes out from the sublingual gland. The sublingual gland is heavily mucosal. Mucus is a type of saliva that's high in mucins, which is a complex polysaccharide that makes the saliva real slippery and slimy. And then the serous saliva is very watery. The mucosal one is also quite oily, but the serous saliva is very watery in its composition, and that's from the parotid gland. The intermediate gland, the submandibular one, is a mix of those two. All right, so let's see that a little bit closer. What do these things even look like? So here's a cross section of I don't know whether this is actually the submandibular or sublingual. It's one of the two. It's not parotid. And we can see here, oh, this must be submandibular. This is submandibular because it has both serous cells, which are the dark purple, and then the white mucus cells, which are the very light-colored regions in that. And then there are these ducts, these little tubes that transfer the saliva into the oral cavity. This saliva that's produced contains talin. And this is another name for amylase, the same sugar hydrolyzing protein that we've talked about many times already this semester. But salivary amylase, or talin, or tyalin, I don't actually know the right pronunciation there. It's going to break apart alpha-1,4 glycosidic linkages that you'd find in starch. So salivary glands. All right, so then you swallow your food, and it travels down to the stomach. Let's just look at the stomach anatomy for a moment so we can talk about the chemistry of the stomach. So your stomach is really wrinkled on the inside. It has these ridges, which are called rugae, and it allows the stomach to do a number of things. One of them, it allows it to expand and contract. These ridges create these gastric pits, kind of a fun name, little holes that go down into the lining of the stomach. These gastric pits are lined with a number of different cells. The ones that are important to us are there's three that are important to us. The parietal cells. Parietal. Does anyone know what the word parietal means? Not in an anatomical sense, just it has a generalized definition as well. So I went to, you guys have co-ed dorms here at Colby, right? Yeah. I went to graduate school at Notre Dame, which is a Catholic school, and there they don't have co-ed dorms, not surprisingly. They have these things instead called parietals, and a parietal the word parent is related to it, but it means wall. Parietal means wall, and a parietal there was 10 o'clock. All the boys had to get out of the girls' dorms at 10 o'clock and vice versa. And it was this wall on which no one could pass, right? So here it means wall as well. It's just the parietal cells are the cells that are in the walls of these gastric pits. And then the bulk of the cells are these chief cells that you see down there. And at the very bottom are G cells, and G standing for gastric. The parietal, G cell, and chief cells together make up the gastric gland, which is at the bottom of the gastric pit. This is where we're going to make the stomach juice that helps us to digest our food. And we'll see how that works in a moment. All right, so here it is. This is a kind of an interesting story. So there's a picture of a gastric pit. Gastric juice that's produced in these gastric pits has a really high concentration of hydrochloric acid. And hydrochloric acid is quite acidic. It's a strong acid. So the pH can be as low as 0.8. That's very acidic. but specifically where this comes from and how it affects the rest of your biochemistry is kind of an interesting story, to me at least. So here, it's produced by the parietal cells. Here on the right-hand side, you see a depiction of the parietal cells. And on the right, in the sort of rose-colored area, that's the inside of the gastric gland, which would lead out into the stomach. All right. And on the other side, in the blue, there's this interstitial fluid, which is how materials would get into the bloodstream. All right. And in between are the parietal cells. Well, what parietal cells do is they take carbon dioxide and water, and they use an enzyme called carbonic anhydrase, and they make carbonate. And carbonate, or I'm sorry, carbonic acid, pardon me, not carbonate, carbonic acid. Carbonic acid dissociates into bicarbonate and H plus ion. All right, so it's an acid is a proton donor. The carbonic acid that was made by the combination of carbon dioxide and water dissociates into the bicarbonate ion, which is alkaline, and the H plus ion, which is acidic. And we're going to export that H plus ion into the stomach where we need the acid. But we need a counter anion. You can't just like pump hydrogens across there without balancing the charge. So we need a counter anion, and that counter anion is going to be a chloride. So we're changing carbonic acid, which is a weak acid. It's the acid that's acidifying the oceans, for example, to hydrochloric acid, which is a strong acid. We're doing this simply by switching the anions, the negative counter anion to the H+. We're doing that by this alkaline tide. We're pulling bicarbonate out of the blood, and we're shoving chloride into the blood. So there's an equal exchange of anions. There's a charge balance there. And then that's pumping acid into the stomach. So if you think about what's happening here, we're making the interstitial fluid and thereby your blood more alkaline or basic, raising the pH, while we're lowering the pH of your stomach. All right? So we're pushing acid, we're moving acid essentially from our blood into, the net effect is to move acid from our blood into our stomach contents. All right? So has anyone here, so I know I have a few athletes in the room. Have any of the athletes or non-athletes, anybody in the room, exercised, and I say run because my experience with it has been running. Have you ever run so hard that, and you're like breathing so hard that you vomit? Does that ever happen to anybody? Okay. Why does that happen? Why does that happen? What do you think?
Isn't it because your body, there's too much lactic?
It's not just lactic acid. That's right. That's a good thought. But it's also because there's so much carbon dioxide in our blood because you're burning carbohydrates so quickly. We haven't talked about, and I'm debating whether I should even go through metabolism now because we're running out of lectures. But when you're burning carbohydrates or fats, you're producing all this carbon dioxide, which goes into your blood, right, as carbonic acid, and it's making your blood more acidic. And you don't want your blood to get too acidic. So when you go into acidosis, from exercise-induced acidosis, you want to remove acid from the blood, and this is a really quick way of doing it. It's just you're rapidly pulling acid out. If your lungs aren't doing a good enough job blowing off all that carbon dioxide that you need to pull out of your blood, then your stomach, this mechanism right here of creating hydrochloric acid, will pull acid out of the blood, making it the blood more alkaline, and then just removing the acid from the body.
So the CO2 acid.
Yes, because carbon dioxide, so in this first step here, you see how carbon dioxide when carbon dioxide goes into solution in water, it automatically, there's an equilibrium where it's going to produce carbonic acid. So carbon dioxide in solution is in equilibrium with carbonic acid. So the more carbon dioxide that your body is producing in the cells that's going into your blood, a lot of that is becoming carbonic acid. All right? And so if you get too much CO2 in your blood, you get hypercapnic. It's called hypercapnia. Your blood you go into acidosis, and it can be really problematic if you don't take care of that quickly. So one of the mechanisms is just elimination. Yeah, it's well, it should. That's physiology for you. Okay. Any other questions on this? So let me think. 20 minutes. I want you guys to have 15 minutes. Okay, so that's how your stomach produces acid. There are other proteins. So an important protein that your body makes is called pepsin. And pepsin is made by the chief cells. So I told you that the parietal cells make HCl. And the chief cells are the source of pepsin, but they don't make pepsin. They make something called pepsinogen. Pepsinogen is a proenzyme. Another word for that is a zymogen, Z-Y-M-O-G-E-N. What an amazing Scrabble word right there, zymogen. Put that one in your word bank for Scrabble for Thanksgiving here. A zymogen is an inactive enzyme. It has not been activated yet. It has a regulatory peptide that's attached to it that prevents the peptide from properly folding. Or maybe that regulatory peptide in this diagram, the orange chain, maybe it's blocking the active site. Whatever it is, the protein can't do its enzymatic job properly until it's been removed. Pepsinogen, turns out, not surprisingly, is easily hydrolyzed. The peptide is removed by HCl that would be produced by the parietal cells. So the parietal cells start to pump out HCl. The chief cells pump out pepsinogen, which is not active, but when it comes in contact with HCl, boom, suddenly the peptide gets removed, gets chewed up, and then the pepsin itself is active. Pepsin is an enzyme whose job it's a protein whose job it is to break down other proteins. It cuts them up. And so all the dietary proteins, that delicious tofu burger or rare bloody steak, whatever you ate, that dietary protein that's in there is going to get chopped up into smaller peptides, smaller chunks of amino acids and small chain peptides by pepsin. All right, that's how pepsin gets activated. All right, so let's go through the phases of digestion. There are 3 phases, and on the left, I'm going to go through the sort of detailed chemical sequences, and on the right, I'm just going to give you some easy to relate to steps in the meal. All right. So the first stage we call the cephalic stage. Cephalus. Does anyone know what the word cephalic means? It does not mean phallic. It means cephalic. The cephalus is your head. All right. This is the part of digestion that's all up here, folks. What do you smell? What do you see? What does the food taste like in your mouth? My gosh, that stuffing looks so delicious. I'm just thinking about it now, man. I'm turning my stomach on just with thoughts of it. This is the cephalic phase, and as you do this, the vagus nerve, which is one of the cranial nerves, in fact, it's cranial nerve 10. That's what that NX up there means. cranial nerve 10, the vagus nerve, sends signals down to the stomach. The word vagus is related to the word vagrant because vagus is the wanderer. It's the wanderer. It's the wandering nerve of the body. It wanders throughout. It goes all over the body, does all sorts of things. It goes to the lungs, the heart, the stomach, the guts. It serves all sorts of viscera. But this branch of the vagus nerves goes to the stomach and starts turning on all of the different cells. The parietal cells, the G cells, the chief cells. There's some mucosal cells that I didn't talk about that are going to make mucus. They're going to put fluid into the stomach. The mucus cells make the mucus. The chief cells produce pepsinogen. The parietal cells start making HCl, which is going to activate the pepsinogen. And then the G cells make this enzyme called gastrin, which we will get to in just a moment. All right, so this is before a single lick of food goes down the hatch. All right, this is the cephalic stage. So the totally just central nervous system turning things on. Step 2, we're actually eating. We're putting food in our mouths and swallowing it. Our stomach is getting full of food. So a couple things happen here. There are sensory nerves in the stomach that are stretch receptors. And when the stomach begins to stretch, they turn on. There are also chemoreceptors that tell when the pH, that says elevated pH. That shouldn't be elevated pH. That should be lowered pH, elevated H plus ion. That's a bad diagram. I'll have to fix that. But as the pH lowers or gets more acidic, it turns on these chemoreceptors. These cells, they don't even go up to the brain and come back down. These are local nerve cells in the enteric plexus, the nerve system just of your GI tract. And they turn on, they reinforce the signals that the vagus nerve was bringing down to the stomach. All right, they continue to turn on these 4 cells that I talked about. So as we have some partially digested peptides, the partially digested peptides further stimulate the G cells to produce a lot more of this gastrin. Gastrin does a couple things. It further turns on chief cells. It further turns on parietal cells, making more pepsinogen, making more HCl. This is an example of positive feedback homeostasis. So this is an accelerating process here. It also causes your stomach to do this, the churning motion. Your stomach is basically a muscle that's going to mix the food all around. All right. And then the last phase is the intestinal phase. This is when you feel full. Okay. And essentially what this does is there suddenly becomes, instead of just like churning waves, there's this peristalsis, like a coordinated motion of the stomach that's going to shove the food out of the stomach and into the intestine, where there's going to be a whole host of these enzyme, these hormones like cholecystokinin, gastric inhibitory protein, secretin, that are all going to turn off everything in the stomach. Their job is to turn off the lights in the stomach, shut down the chief cells, shut down the parietal cells, shut down peristalsis, shut down the myenteric plexus. Okay. Steps 1, 2, and 3 in digestion. Okay, so now the food is in the duodenum, and let me see. I've got a little over 12 minutes here, 14 minutes. I'll talk about this guy and then we'll break. So peptic ulcers. Peptic ulcers or stomach ulcers, but when I was a child and you watch these like TV shows about cops driving around in muscle cars, throwing hot dogs out of their window, answering the call to catch bad guys like Starsky and Hutch or something like that. You'd get the police chief who had a nasty, he was getting a gastric ulcer because Starsky or Hutch had just wrecked or blown up another building or something like that. And he had to lay off the hot dogs and donuts. That is not actually how peptic ulcers work. And as you guys were kids, hopefully, I wasn't really watching TV, but maybe as you were, you didn't see any caricatures like I just described for you. And that's because of this guy. So this guy, Barry Marshall, way back in the 80s, came to realize he was culturing the bacteria that lived in people's stomachs, and particularly culturing the bacteria that were in people's ulcers, in gastric ulcers, peptic ulcers. And he found this one bacteria, Helicobacter pylori, was way overrepresented in peptic ulcers. In fact, he believed that a peptic ulcer was essentially a runaway infection by this particular bacteria, H. pylori. And people told him he was crazy. It's just because there's too much stomach acid, you're eating food, the wrong kinds of foods, greasy foods are going to give you a gastric ulcer, foods with a lot of meat in it. And he didn't back down. So he had papers rejected. People told him he was crazy. He'd go to conferences and present, and he would essentially be scorned and ridiculed by his peers. But he stuck to his guns, and he was determined to prove them wrong. So what would you have done in this situation? He made a big old beaker of the stuff and just drank it down. Gave himself a wicked case of ulcers, took a course of antibiotics, cured it. 24 years later, Nobel Prize. So there you go. Kind of an interesting story. Okay, so I'm going to stop there with this.